Hello everyone, and welcome to today's keynote presentation, The Invisible Influence of the Microbiome, presented by Dr. Jack Gilbert. We are excited to bring you this educational web seminar presented by LabRoots. For more information about LabRoots, please visit www.labroots.com. I'm Bob Woodard of LabRoots, and I'll be your moderator for today's event. Before we begin, I'd like to make a few announcements. First, this is an interactive event, and we encourage everyone to submit questions and comments during the presentation. To do so, click on the Q&A button in the lower left of the presentation window and enter your questions and comments in the Q&A box that appears on the screen. Also, the slides will be presented in the slide window of the screen, and you can enlarge the slides by clicking on the Enlarge Screen button in the lower right of the screen window. Finally, if you have any difficulty viewing or hearing this presentation during the broadcast, please click on the support button in the upper right of the presentation window, or let us know what the problem is using the Q&A button on the lower left. With that, I'd like to introduce today's speaker, Dr. Jack Gilbert. Dr. Gilbert is currently applying next-generation sequencing technologies to microbial metagenomics and metatranscriptomics to test fundamental hypotheses in microbial ecology. He has authored more than 120 peer-reviewed publications and book chapters on metagenomics and approaches to ecosystem ecology. Dr. Gilbert has focused on analyzing microbial function and diversity, community assembly processes, and dynamic interactions between taxa with an aim of predicting the metabolic output from a community. He is currently working on generating observational and mechanistic models of microbial communities in natural, urban, built, and human ecosystems. He is on the board of Genomic Standards Consortium, is a section editor for PLOS One, and senior editor for the ISME Journal and Environmental Microbiology, and is PI for the Earth Microbiome Project, Home Microbiome Project, Gulf Microbial Modeling Project, Hospital Microbiome Project, and the Chicago River Microbiome Project. Please join me in welcoming Dr. Gilbert to today's presentation. I will now turn the presentation over to him.
Right. Hello, everybody. It's great to be here. My name is Jack Gilbert, and as Bob said, I'm an associate professor in Department of Ecology and Evolution and Department of Surgery at University of Chicago, as well as being a group leader in microbial ecology at Argonne National Laboratory and a senior scientist at the Marine Biological Laboratory over at Woods Hole. I am very excited to be a microbial ecologist in this day and age. It's a fun thing to do. We're starting to understand that microbial ecology has a significant role to play in understanding all of the aspects of ecology that we see in various environments. I'm going to be talking today about the human body, but ostensibly the human body is no different to the ocean or a soil or a plant or an animal. It's just another environment, another ecosystem that's been colonized by bacteria. And these bacteria have found ways to adapt and to interact with the human body in many intricate pathways. You have around 100 trillion bacterial cells in your body, and everyone bands around the number 10 to 1 in terms of the 10 bacterial cells for every one human cell, but it's really more like around 2 to 3 times the number of human cells in bacteria. So they do weigh around 2 to 3 pounds, which is about the same weight as your brain, so they're potentially very important. The vast majority of them reside inside your gut, but even your skin is intricately integrated into microbial ecology. There are bacteria from the surface deep down into the sublayers, and they inhabit very, very complicated niches throughout that system. I'm going to be highlighting work today on helping us to understand how those bacteria interact with our body with regards to their interaction with our neurological behavior through the immune system, our response to food allergens, our response to food in general, our neurological development, and even our ability to gain weight. And then I'll finish up by talking about where these bacteria come from and how we interact with them in our modern world. So I'll start off by saying we need microbes. They're very important. Bacteria and viruses are needed by the body in order to allow the body and brain to develop properly. In this image, we can see three different mouse seeker. This is a part of their gut that contains a lot of the bacteria. The one on the far right, the tiny little squiggly one, that's a healthy one. And the one on the far left is an unhealthy one from a germ-free mouse. Or you could call it unhealthy. It's a germ-free mouse, as long as you don't take it outside of its bubble, is perfectly healthy. But it's biologically very different from a mouse which has bacteria associated with it. As you can see, the physiology of its cecum is changed dramatically. It's enlarged, it's inflamed. The immune system is responding to the absence of those microorganisms by altering the development of the physiology of the mouse. And this is because organisms, you, me, mice, plants, animals, they've evolved in a sea of microbes. They never evolved a way to eradicate or get rid of all those microorganisms, so they've evolved to deal with them. One of the ways they've evolved to deal with them is to develop a complex immune system that helps them to interact and even utilize those organisms to develop their physiology and to interact with the world. We know we need them. We think that human social behavior and the social behavior of many organisms may have evolved to allow their propagation between animals. In a fantastic piece of work from Duke University, Tung et al., they explored the distribution of microorganisms through baboon populations, and animals that frequently groomed each other were more likely to share bacteria than animals that didn't groom each other. And this sharing overrode any different response to food intake. So diet didn't significantly have as big an impact as the ability of those animals to groom each other. And therefore, even human behaviors such as shaking hands or kissing or hugging may have evolved to propagate the distribution of bacteria within a population of humans. It sounds crazy, but it really isn't that crazy. On that balance, many of our behaviors may be influenced or may have evolved to enable us to either avoid bad organisms or promote the distribution and acquisition of good organisms, which help our body to respond appropriately to different environments. Now, when we consider that relationship with behavior, work by John Cryan, for example, has highlighted the extreme importance of the gut-brain axis, the relationship between what happens inside your intestine and what's going on in your brain and your behavior. We know that there are many different mechanisms by which microorganisms can influence neurological behavior. So, for example, bacteria can influence interactions with the immune system. The immune system can change inflammatory status. This can affect neurological firing through the vagus nerve back up into the brain. Bacteria in the gut may affect myelination in various parts of the body, and therefore diseases that are due to a reduction in the amount of myelination may be altered by the type of bacteria that are present there. In this particular study we're showing here, Cryan and many other colleagues have explored the role that bacteria in a mouse plays in its behavior. So we know that when you take a germ-free mouse, a mouse without any bacteria, it exhibits a behavior which is very bizarre for a mouse. A mouse should be highly afraid. It should be very anxious of its environment, because the brave mouse that wanders out gets eaten very quickly, and the anxious mouse that hides away from predators remains alive and breeds. But when we take bacteria away from a mouse, the mouse becomes very brave. It reduces the anxiety that mouse experiences, and in this instance we have an elevated maze. The mouse that's inside that maze will come outside of the box if it doesn't have bacteria and walk along this elevated platform as if everything's fine. And when it has bacteria inside it, when it's grown up in a conventional way, the mouse will stay hidden inside the box and will prefer not to walk along that platform. And that's very important. So the bacteria in the gut can alter the behavioral abnormalities or behavioral physiologies of an animal at very specific levels, and therefore it can do the same to us. We also know that bacteria can have a big impact upon how we respond to different foods. So problem foods like peanuts and shellfish and milk cause many. 32 million people currently suffer from these kinds of allergies, and they cause a histamine response and can lead to hives and massive inflammation and sometimes anaphylactic shock. And so it's very important for us to understand the potential role that bacteria could be playing in the propagation or the substantial reduction in the prevalence of food allergies. We know from work with Kathy Nagler at the University of Chicago that adding clostridial organisms back into a mouse model very early in its development can significantly desensitize that mouse to peanut allergens, so that the mouse will no longer show an immunological response or an inappropriate immunological response to those allergens. And this is very important. We've also, working with Kathy, we've seen the same thing in cow's milk, with cow's milk allergies, which affect a large number of infants and people. We think that the abundance of organisms that can generate short-chain fatty acids, such as butyrate, can play a significant role in exacerbating the desensitization or oversensitization of infants, human infants in this case, to cow's milk allergens. And so working in this allergic space, we're able to explore the role in which bacteria can play in reducing the allergic response to specific foods. We believe that the generation of short-chain fatty acids is very important in this instance, in that these short-chain fatty acids are feeding eosinophils and other immune cells inside the epithelial lining of the gastrointestinal tract, and therefore feeding back to significantly reduce inflammatory status via an IL-22 response. We also know that bacteria can change the way you sense foods. So even as early as food sensing in the oral cavity, in the oral pharyngeal cavity, we know that, for example, wine and the way wine is processed by the saliva and the bacteria in the saliva can affect the number of aromatic chemicals which are released in the mouth and therefore taken up into the nose. In a study with Danilo Ercolini in University of Naples, we were able to demonstrate that obese people, they had bacterial signatures in their saliva which seemed to alter the chemical perception of wine and the nasal pharyngeal perception of that wine by reducing the number of aromatic carbons released during the oral process. And this is important. Well, wine is important to me, but it's also important because it means that as early as food intake into the mouth, our behavior, our response to these foods is already being altered, already being changed by the activity of microbes. We also know that we can potentially alter neurological phenotypes. It's be controversial to say we can cure autism. We can potentially alleviate autistic symptoms in mouse models at this stage. But many other symptoms, such as depression and anxiety, and conditions such as learning ability or the ability to retain information, can be altered by the presence or absence of certain types of organisms. In post-surgical depression, for example, we can see a significant reduction in the incidence of post-surgical depression with the application of simple probiotics, such as Greek yogurt. Now, those studies need to be replicated in a much more robust and double-blind trial way in order to determine the absolute efficacy. But it's an interesting lead in helping us to understand the potential link between neurological depression and the microbiome. In pioneering work from Sarkis Mazmanian and Paul Patterson out at Caltech, they demonstrated that the introduction of a single microorganism back into an autistic mouse model actually significantly reduced the symptoms of autism in that model. This was by the simple introduction of a probiotic, which moved the microbiome in that mouse from a disordered autistic phenotype to a more healthy normal phenotype, which changed the level of different metabolites in the serum of the mouse, the blood of the mouse, and therefore potentially altered the endocrinological, immunological, and neurological behavior. And there is hope to push this work out into clinical trials in order to test its effectiveness on humans. We also know that bacteria can trigger other neurodevelopmental conditions. Parkinson's and Alzheimer's could be altered by bacterial interaction and metabolism in the gut, and as well as bacterial metabolites getting into the bloodstream. The abundance of beta-N-methylamyl-alanine and brain-derived neurotrophic factors and GABA have been linked to, in the serum of patients, have been linked to the onset of Alzheimer's, for example. And we are starting to understand how microbial metabolism may be affecting the abundance of those particular metabolites. What's very interesting is that certain conditions such as Parkinson's may be triggered by an alteration or change in the microbiome. And if we can understand that link, potentially mediated through the immune system, we may be able to prevent the onset of these conditions. We know that bacteria can make you fat as well. And this has been very well publicized through many research groups around the country. One great study from Liping Zhao in Shanghai demonstrated that when a morbidly obese person goes on a significant diet, in this case a diet of traditional Chinese whole grains, they also show a significant shift in their microbiome. In this one instance, in the study with Liping Zhao, the individual weighed 385 pounds. He lost 113 pounds by eating a diet of whole grains. And then this was in about 9 months. And then at the beginning, he had an abundance of a particular organism which was almost absent by the end. This organism was Enterobacter cloaceae B29, which, when introduced back into a mouse model, significantly increases the likelihood that mouse will become obese. It's almost as if the mouse is altering the microbial abundance of that, altering the obesity of that mouse. I had a question from Maria Ivanoshkova. I have apologies if I pronounced your name wrong. Can we prevent the onset of autism with bacteria? Potentially we can. It's an interesting point. The ability to prevent the onset of a disease is only predicated on our understanding of that onset. And many of the autistic phenotype is actually due to developmental regression during development. So neurological pathways are not properly formed. The development is retarded during in utero and in early developmental processes. So it may be that we need to alter the health of the mother or the microbial profiles of the mother in order to have any effect during pregnancy. And that could reduce the onset. We're not sure. I'm being told by the organizers to save questions for the end of the presentation. I apologize. Work from Ruth Lay has demonstrated that we're able to also maybe affect the onset of obesity by reducing the, changing the presence of certain organisms. So in Ruth's study with collaborators in the United Kingdom, she was able to show that monozygotic twins, identical twins, actually share more microbial similarity than dizygotic twins or normal siblings. So there's a certain genetic profile there. But in her study, they had obese twins and lean twins in twin pairs. And the lean twin had an extremely significantly elevated abundance of certain organisms that were highly hereditable from the mother, such as Christianolaceae. And the introduction of Christianolaceae into a stool sample from an obese person significantly reduced the likelihood that a mouse that ate that stool sample was able to gain weight. So in the study on the right, taking stool from an obese twin and giving it to a mouse, or the microbiome of that stool, not the stool itself, that'd be gross, but giving the microbiome of the stool to the mouse significantly increased weight gain in the mouse. By adding Christianicella minuta to that same stool sample, they were able to reduce the likelihood that the mouse would gain weight. So this is the opposite to Enterobacter cloaceae in Liping Zhao's study, where he demonstrates that the introduction of Enterobacter cloaceae increases weight gain. The introduction of this one organism, Christianicella minuta, might prevent or protect against weight gain. Obviously, this is a massive generalization, and a significant research needs to be done to demonstrate the potential of these organisms to have any impact upon weight gain. But what's even more critical is we still don't understand much about the functionality, the mechanism by which these organisms influence it. And work with Eugene Chang and my group at University of Chicago has started to look at this from the concept of ecological periodicity. What I mean by that is the change in the day-night cycle of microbes in our gut and how that can affect our circadian rhythm in our body. We all know when we get jet lag or when we work nights, we feel neurologically different and physiologically different. In fact, people who are chronically jet lagged or people who are suffering from sleep deprivation or working nights are more likely to put on more weight than people who are not suffering from those conditions. And in work from Eugene Chang and my laboratory, we've demonstrated that an increase in dietary fats and sugars significantly disrupts the rhythm or the rhythmicity, the daily rhythmicity of microbes in the intestine. And when that daily rhythmicity, that circadian cycle of microbes in the intestine is altered, it also seems to disrupt the circadian rhythms of genes in the hepatic tissues, in the liver, and also in the brain, which has a significant impact on altering the amount of weight gain in that individual, in this case in mice and rats. So changes in the rhythmicity, the day-night fluctuations in the ecology of bacteria in the gut can affect our ability to put on weight. And this may be a significant mechanism by which microbes alter weight gain in humans. Again, that still needs to be proven. So where do these microbes come from? This is fundamentally interesting. We know microbes can alter our body's immune response, but where do we get them from in the first place? We know from work with Maria Gloria Dominguez Bello and colleagues that the alteration of the microbe starts very early, very young. Babies are normally born sterile, and the first environment they're exposed to alters the types of bacteria that they acquire very early and that stabilize through their first year of life. If you're born via vaginal delivery, you look like your mother's vaginal microbiota, and you're colonized by those organisms even in your gut. And if you're born by a cesarean section, you acquire microbes from the skin of the first person you come into contact with. And those microbes colonize inside your gut. And obviously only the ones that can handle the anaerobic environment and the pH variance, but that's an important stepping point. And we know that microbes in breast tissue and breast milk can have a significant impact. And evidence is mounting that the mother may actively recruit bacteria into the breast milk to act as a probiotic and a symbiotic, so a pre and probiotic combined together to provide baby with the right kinds of fats and carbohydrates and lipids and bacteria to train its immune system to respond appropriately, as well as to provide an appropriate digestive capability for some of those milks and sugars and fats that are inside the body. So this is very important to understand how the mother's system and the way you're born and the way you're brought up may affect your immunological status. It's not the only component, though. In a study with Carol Ober, our group is looking at how the experiences you get early in life may affect your development of autoimmune conditions and conditions such as asthma. When we look at two groups in the US, the Amish and Hutterites, we see a significant differentiation. Amish groups in our population of colleagues, Amish colleagues in northern Indiana, have a significantly low level of asthma, less than 1%. And this differs from their Hutterite colleagues in North Dakota, who have a very similar genetic lineage, but those Hutterite populations have significantly elevated levels of asthma, up to 30%. And the average for the US is between 8 and 9%. And what we believe this is due to, remember all of these people are vaginally delivered and they're all breastfed, is early experiences, experiential microbial learning of your immune system. The Amish children are pretty much brought up interacting with animals and with the farm environment, so that they get a very rich contextual environmental microbial experience from very, very early in life. You're bound to your mother as she's milking the cow very early in the morning. You're given mucking out duties. There's really no play outside, inside the house. You are spending a lot of time on the farm. Hutterite children are not treated this way. In the Hutterite communes, the children and parents, mothers, stay in the house, and it's only the men that are allowed on the farm to work in that environment. And it alters the profile of their microbiome because of the experiences, environmental experiences they have. And so it may be that that plays a role in the development of asthma in that population. Of course, this remains to be seen and proven, but it's highly likely that that environmental experience plays a significant role. And this is a very important component of our research in trying to understand how our environmental exposure affects our development of our immune system and our microbial cells. In work with the Alfred P. Sloan Foundation, we performed a study called the Home Microbiome Project, In this study, we looked at people who were moving home and how their microbiome and the microbiome of their home was altered by this interaction. We know that families leave a microbial signature in their home very rapidly. So the vast majority of the directionality of the microbial exposure is from the person into the home environment. And this alters the microbial environment in specific ways. So that if you bring a baby into this environment, the baby is exposed to the skin microbiome of the people or the occupants of that space. If you have a different occupation, you have a different microbiome. And this could be playing a role in the development of certain immune conditions. We know that we can actually use this in a forensic angle. It has nothing to do with immunology, but it's very interesting that I can tell if two people in a space are physically interacting, and you can tell which ones aren't physically interacting. In this case, we had a young couple who lived together who were physically interacting, and their microbiome was significantly more similar than their lodger who wasn't physically interacting with them. This is leading to an entire new field of microbial forensics. In an article in Time magazine last week, we demonstrated that it was possible to identify criminals in an environment at a crime scene by looking at the microbial signatures they've left behind in that space. And we could also identify significant components of their personal life, such as whether they took, in the case of this study, whether they took migraine medicine. So from the microbial profile they left behind in the environment, we were able to predict accurately that one of the criminals suffered from migraines. And this is very important because it shows the potential of these techniques investigating microbial interactions with our environment in other areas of research. We use this information to map and model microbial profiles. This is a young family. It's my family. And we have 2 adults and 2 children. And their feet are microbially interacting with the floor, and their hands are microbially interacting with doorknobs and light switches. And you can see, in fact, nearly everybody's hands also has interactions with the floor, but this one individual in the second hand up, if I can turn that into a whiteboard, there we are. So this individual here has a significantly greater microbial interaction with the bedroom floor. This individual is my young son, and so he was crawling around on the floor and much more physically interacting with the floor space than any of his parents here or his elder brother. That altered his microbial experience. So how you behave in an environment changes the way you experience that environment. This is my son, my elder son, microbially swabbing himself, so you can get your family involved. When you add dogs into an environment, it significantly alters that microbial profile. This is a family with dogs, and you can see that the complexity of microbial interaction spaces and the breadth and width of those interactions is significantly increased. Because dogs run around more inside the home and interact physically more with other people. In fact, work from Rob Knight's group in University of California, San Diego, and University of Colorado demonstrated that the microbiome of people that owned a dog was significantly more similar than people who didn't own a dog, because the dog was acting as a conduit to increase microbial sharing between those people. This is important because dogs are awesome. I actually, on the basis of that research, rescued my dog, Captain Bo Diggly, to significantly increase microbial sharing in the house in a totally unscientific idea that the dog might improve our microbial profile and then maybe improve the immune health of my family. Maybe not entirely unscientific. Work has demonstrated that bacteria from dogs can protect against asthma development in a mouse model and asthmatic symptoms. In fact, particular lactobacillus from dog feces may play a significant role in reducing the prevalence of asthmatic attacks in this mouse model, and therefore it could be the same for humans. I'm very keen to explore the potential to put these microbes back in your medical records so that we can maintain a fundamental understanding of how your microbiome is altered and how it interacts with you through your daily routine. So the only way to do this really is to either install implants in our body that monitor the microbial health of different components of our body, and we're a long way off doing that effectively, although there are methods being developed to achieve that. The other way is to look at your stool each day, we are in the process of developing a microbial sensor which has the potential to monitor different microbial organisms in stool samples as they pass through a toilet system. And groups as far as wide as MIT and New York researchers are trying to explore the potential of bacterial microbial signatures in our wastewater systems as potential indicators of human drug-taking activity or human health activity in large urban environments. In fact, Eric Alm's group from MIT did a great study where they explored the microbiome of two individual men every day for a year. You can see immediately in this instance that the top profile is significantly different from the bottom profile. This is because there are two individual people with two unique microbiomes. But interestingly, you can see two weird instances in this. Let me highlight it again. Up here, you see a significant increase in Proteobacteria at one particular time point during the year. And this is when one of the individuals got food poisoning, a salmonella infection, which did actually go away and they got better. But their microbial profile, while it looks very similar to what it was before, there was actually significant differences between before and after the infection. Here, on this other individual, they got an increase in Proteobacteria, which also includes salmonella, when they were living in Bangkok, Thailand for a month and a half. And this significantly altered their microbial profile. But interestingly, as soon as they returned to the US, their microbial profile got significantly more similar to what it was beforehand. So this is important. Over the year, their microbial profile is quite stable, but there are specific instances when it changes. And what if a drug that the individual number 2 wanted to take at this time point was less effective because of the alteration of his microbial profile based on where he had moved to? And his drug response may be changed, and that may significantly alter that person's response to that drug, which will change their health outcomes. So understanding this could have potential implications for treatment strategies. In a pioneering study we've been doing in a new hospital on the University of Chicago campus, we've been exploring how the patients and staff of that hospital environment microbially interact with their space. And my student Simon Lax has been exploring this to determine when the hospital became active. We got very lucky. We had a $700, $800 million hospital built on our campus, and we were able to analyze the building before any of the patients or doctors moved in and look at the bacteria that are present there, and then look at the bacteria that changed in the environment and in the patients when the building became operational and occupied. And so occupancy, in blue and white here, we have microbial profiles from environments prior to opening, and in red we have microbial profiles from environments after opening, was significantly altered, significantly changed by the presence of all those people. And that's very important. And we're learning a lot about the succession of the bacterial profile in that hospital environment and what impact that succession could be having upon the spread of antimicrobial resistance and also on the development of potential healthcare-associated infections in that ecosystem, with the potential ability to change the hospital environment to alter it for the better. We've even gone into restrooms this is a university, this is San Diego State University, and looked at the types of bacteria which are present in that restroom and how they change over time. So we can detect how different cleaning strategies may affect it. So at 7 o'clock in the morning in 6 restrooms in San Diego State University, we went in and we sterilized them for 20 minutes with bleach until there was nothing there, all the organic material, DNA and RNA being completely removed. And then we swabbed the surface every hour from 8 a.m. We barcode that from 8 a.m. here until 3 o'clock in the afternoon. And we saw significant changes in the microbial profile over that time. And you can see that the bacteria that predominantly come from stool are significantly reduced over that time frame, and the bacteria that come from skin are significantly increased over that time frame. And that reduction and increase is due to the successional change in structure and response of the communities in that environment. And we can see that many of those organisms are still biologically active and are responding to the interaction with that space. And if we look at those environments every day for a week or a month, we see that the microbial profile every day for a month on average looks very similar to what happened after 8 hours of microbial succession. So if you don't sterilize these floors, they reach a stable state. And that stable state is potentially has less gastrointestinal enteric organisms than it does skin organisms, and therefore could potentially be slightly healthier, but that's to be determined. The soap and toilet seats, however, always maintain a very high level of stool-associated microorganisms, which is probably not surprising. In collaboration with Rob Knight and Jeff Leach and many other colleagues across the US, UK, and Australia, we've developed an international study called American Gut, or British Gut, or Australian Gut, that allows people to look at their microbial profile for a small fee. And all of this data then is made immediately public, so that all researchers around the country and anyone else can have a look at microbial profiles from a very large cohort of people. This is important because it enables us to explore microbial diseases, microbially associated diseases, in a very large portion of the population. By looking at a small clinical study and looking at the microbial organisms which are important for that study, i.e. maybe disease-causing or disease-protective, we can then go into this large cohort of 5,000 to 8,000 people now and explore the prevalence of those organisms across a much larger population, and a population that's told us about their disease burden, their diet, and their age, and their gender, and their lifestyle. And that helps us to explore in a data correlative way the potential implications of those organisms in a larger population. In this instance, through using deep genome sequencing, we looked at a husband and a mother and a daughter and a son in the same family unit, and reassembled the genomes of all of the organisms present in each one of them, and looked at the co-association of those organisms between the individuals. The son had crippling allergic reactions and allergenic responses, and he shares a particular E. coli strain with his father, but not with his mother or his sister. And there are also other organisms present here which are highly indicative of the son having a differential microbiome to either his father, his mother, or his sister, which could play a role in the onset of his allergic condition. Again, that needs to be proven. I'll end the presentation there, and I'd be happy to take any questions. I have questions.
Well, thank you, Dr. Gilbert, for that excellent presentation. Now it's time for Q&A, and if you have a question you'd like to ask Dr. Gilbert, now's the time to do it. To do so, click on the Q&A button in the lower left of the presentation window and type your question into the box that appears on the screen and click on the Send button. Okay, our first question, we'll answer as many questions as we have time for, or as many as we have. So we'll get started. We have one question right now, and that's from Laura Echeverri from the Universidad CES. The question is, in this order, we can say that genetics and microbiome alterations lead to metabolic alterations, therefore predisposing to certain diseases. Cardiovascular diseases are the first cause of death worldwide, and the result of many factors, among them obesity. So is it possible that cardiovascular disease can be improved by detecting a fail in normal microbiome activity and restoring it?
So, yeah, am I online? Yeah, I have full control. So this is a very important question, Laura. Yes, it's entirely possible, and we know that bacteria in the gut can alter through their own metabolism of food that we consume, can increase the incidence of metabolites which can have an impact on cardiovascular disease, such as carnitine in our bloodstream, and therefore, by proxy, through their metabolic capabilities, they're able to alter it. But it's highly possible that we could improve the detection of the onset of cardiovascular disease by looking at how the bacterial community is changing in individuals. So I can see a point in time when the microbial profile of individuals would become a part of the normal of the normal checkup of people as they go through their day-to-day life. Ideally, I would like to see it done very, very regularly, and therefore put the information in the hands of people so they can analyze it more regularly.
Thank you, Dr. Gilbert. Our next question is from Lauren Regman at the University of Washington Medical Center. In the example you gave regarding people's microbiomes being more similar when living together or when with increased contact, do you think bacteria could play a role in why women's menstruation cycles sync up when around other women?
Yeah, am I on? Okay, so yes, actually, it's a possibility. There's no evidence to suggest it's a definite possibility, but when we look at animals who are cohabiting, many of their physiological and neurological processes tend to sync up around the exact same time that their microbial profiles become more similar. And so it's possible that, that alterations in the microbial profile could be leading to shifts in the endocrinological, immunological, and neurological systems in the body, which may lead to a significant, increase in similarity of those responses across individuals. Of course, for the syncing up of menstruation, I don't know. I could hypothesize a number of mechanisms by which that would occur, and I don't know if any of them are real, but it's always a possibility.
Our next question from Alberto Mendoza of the Instituto Politécnico Nacional. I'm not sure, I'll read the question as it stands here, and it says, I wonder why daughter has not few interact with Proteobacteria.
So this is an interesting point. You know, often we see a significant increase in Proteobacteria as being a potential indicator of a detrimental or less healthy, in inverted commas, microbiome. And that's a very, very broad statement. But it's interesting to note that the daughter had no significant allergies, and the son had very crippling allergies, and the mother and father had very, very mild allergies. And the son's, it has a significant increase in his proteobacterial content. Now, Proteobacteria is like saying, you know, it's like saying there are lots more, lots more insects in this ecosystem than animals, you know, mammals. It's a very, very, very broad definition of many, many, many thousands of possible species. So that's why it's very important to get down to the nitty-gritty and examine the genotypic diversity of the son and daughter and determine which of those organisms are relevant. And we've started to do that with that study.
Well, thank you. Our next question from Maria Ivanovska, Medical University of Plovdiv. What about microbiome and immunological reactivity under acute or chronic stress?
Okay. So I actually see two questions here. I see one from Mohammed El Zawati from Emory, as well as the other one. The other one's just disappeared from my time frame. Oh yeah. So I'll answer the one from Maria just now. So what about the microbiome and immunological reactivity under acute or chronic stress? So acute and chronic stress does significantly alter microbial profiles. And in the few studies that have been done, we don't know whether it's cause or effect. We believe that the immune system is obviously responding to chronic stress and therefore altering the ecological environment of the gut and therefore changing the bacterial community. But it's a positive mechanism. There's a dynamic mechanism between those two components. When the microbiome changes, it also changes the immune response. So the immune response changes, it changes the microbiome. So there has to be a bidirectionality there that needs to be understood. And chronic and acute stress are two key components leading to the onset of depression and anxiety. And therefore, understanding of the triggering mechanisms and the link between stressful situations, alterations in microbial profile, immune response alterations, endocrinological response alterations, and then that final tipping point to a disruption of normal neurological behavior has to be better understood. And there are many groups around the US trying to look into that now. The one from Mohammed suggests how to make use of the HMP, Unassumed Human Microbiome project at the moment, and what is the current output of such research? Have you looked at the virome and microbe? I wonder, Mohammed, if you're talking about the hospital study. the output of it at the moment is helping us to understand disease spread within a population, and to track the microbial profiles of people as they go into a hospital, as they leave a hospital, so we can determine points at which they might acquire organisms which could be detrimental to their health. And we have looked at the virome and the mycome, but those results are interestingly variable. And so we are exploring those at the moment. Bob, do you want me to just keep on going? I can't hear Bob. I'm assuming everyone can still hear me. So if... The
next question is from Russell Higbee of the Sanofi Pasteur Vax Design Corporation. His question is, very nice presentation, Jack. Do you think that individuals who learn what their microbiome may contain, and knowing their disease burden, familial history, they could alter their own microbiome over the course of time that would substantially reduce the risk for any given disease they wish to avoid?
Absolutely. Yes. I mean, it's another component of your body's system, right? The bacteria that live inside us, they are intricately associated with us. So by understanding them and how they change, we may be able to alter, and I emphasize we may be able to alter the onset or the problems of certain diseases. I mean, it's very important for us to be able to understand what's the mechanism behind these alterations and changes. But as well as understanding your genotypic history, your familial history, your drug history, your diet, as well as understanding your response to environmental allergens, the microbiome is just another component of information we can use to effect a more personalized control of our medical treatment.
Excellent. Next question from June Kim of Apollonia Health Incorporated. In your research, has any evidence of inter-microbiota communication or interaction been found, for example, between oral microbiota and gut microbiota, or skin and oral?
Yes. So obviously between oral and gut, quite common. The stuff passes through the oral cavity, and any bacteria washed down with saliva into the esophageal tract enters into the GI tract. If it can pass through the stomach acid, then it will obviously enter in and has the potential to colonize other areas of the gastrointestinal tract, but skin as well. So when your skin microbiome is associated with what's going on inside you and what's going on inside your mouth, the very act of you're touching your mouth or your lips with your hand is transferring microbial signatures. And, obviously we use our hands to clean ourselves up after going to the toilet, and therefore there's another route by which microbial transfer can occur. This has been known for epidemiological studies for many years as a potential route to fecal oral contamination. But it's also a route for other organisms which could be beneficial to transfer around our body. It's just the fact that the different parts of our body, the different ecosystems, the skin, the mouth, the armpit versus the knee, the stool, the vagina, the penis, etc., have very different ecosystem properties which shape the types of organisms which can bloom there and can stay there.
Our next question from Marie Young of the California Polytechnic State University. How reliable is the database to ID genus/species? Did you see many unknown species in the samples?
Yes. There's a misnomer between our ability to taxonomically identify organisms down to the genus and species level with a high accuracy and our ability to understand their functions. We are very, very good at identifying the type of genera which are present, especially in human microbiome samples. We know a lot about the genotypes of organisms which are present there. And in many cases, especially with the study that I presented with the family in American gut, we're able to identify these things down to the strain level. So we know that the different strains of species which are present in that sample. We don't see a lot of unknown organisms. Most of the time, using a 16S ribosomal RNA strategy, we can identify 99.9% of all the organisms. If you were to take a metagenome of one of these samples and to try and predict the number of the diversity of the species, or try and identify the idea of each of the species based on their functional genes, then you'd suffer a problem because most of the functional genes are not well annotated. So you get a lot of unknowns. But at the 16S level, and for genotype level, where you have those phylogenetic markers, you can very easily get down to a very fine taxonomic resolution. But yes, you always see some unknown organisms, but mostly they're just relatives of things that we do know a lot about.
Next question from John Inadomi of the University of Washington. What is the mechanism by which alterations in the microbial ecology influence immunology, endocrinology, and physiology? In other words, what are the bugs doing?
That's a very big question, John, but let me give you an example. So we know that microbial organisms can generate metabolites such as hydrogen sulfide, which have a significant likelihood of increasing inflammatory status around those organisms. So they release hydrogen sulfide that causes inflammation in the gut, which obviously affects immunological, is in and of itself an immune response to stimuli. The presence of those organisms elicits an immune response through the release of IgA, which is labeling organisms which could be potentially pathogenic or dangerous, but also keeping organisms structured in the gut and away from the cells of the gut lining, and keeping them at the surface or near the surface of the mucosal layer. So there's a lot of opportunity for those organisms to interact directly with the immune system and therefore alter the immune responses around the rest of the body. We know, for example, that by adding certain organisms back into a rat model, we can actually induce systemic, so body-wide immune responses in the rat model. So we can actually, if there's an infection in the liver, we can change bacteria in the intestine, which will elicit an immune response, which is also elicited in the liver and then clears out the infection in the liver. So, and this may be a way in which fecal microbiome transplants have a significant role to play in altering systemic health. Other ways are through changes in the nerve impulses up from the vagus tract, and that's through cytokine responses, so again immune responses to microbial communities present in the intestine, and also through the release of microbial metabolites into the serum, which can influence endocrinological functions from glands, but also through enzyme degradation rates in the liver. So there are many, many routes by which things are connected, and that connection is very important.
Well, if there are no more questions, I'd like to once again thank Dr. Gilbert for his presentation. And Dr. Gilbert, are there any, do you have any final comments you'd like to make?
So thank you very much for listening, and there are some great questions. Thank you, Shazia, for learning a lot. I appreciate the comment. You know, I gave a very, very high-level overview of some of the research that's going on. I strongly recommend people who are interested. There are many good review papers out there to get integrate into understanding the various components. And this is a hot topic right now. So a hot topic comes with a very specific burden. And that burden is education and understanding of what can and cannot be done. The microbiome is not the be-all and end-all. It's definitely not the mechanism by which we're going to save the world. But it's a component of our health which we ignored for a while, and now we're not ignoring it. So it's very important that we take that on board and understand it effectively.
Well, thank you once again, Dr. Gilbert, and thanks to our audience for attending today's presentation. I'd also like to thank our sponsor, LabRoots, for making today's presentation possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing for 6 months from today's event. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.